If you say target ID is the first step, then the next step should be target validation. In target ID, only we can tell whether that specific protein is specifically binding to your small molecule, but it does not tell you whether this protein has relevant connection to your active molecules. For example, let's say we have seen this cell cycle diagram before, and if one cell is going into the cell division, they need to go to G1, S, G2, M different stages. Preparation here, and DNA synthesis, and another preparation, and cell division happens. And many proteins are involved here. CDK2 is involved in early stage checkpoint here, and CDK1 in a later stage of checkpoints. These proteins are very important checkpoint for cell division. That's why these CDKs are very important target for anti-cancer drug development. But these two proteins are evolutionarily connected to each other very closely. That's why structure is also very similar. It can be quite difficult to develop selective probes for only one of them. That's why if you develop CDK1 or CDK2 inhibitor, in a later stage, you need to confirm those selectivity or specificity against one of those proteins. If you do in vitro screening, let's say, so in this case, you take CDK2 as a purified protein and then screen out different library compounds, then most likely your first screening is one concentration and measuring those kinase activity. So by looking at this table, and if this is 100% of a kinase activity, among these compounds, can you tell which compound is more active than the other? For example, the high bar here versus the low bar here, which compound is more stronger inhibitor to CDK2? Can you tell? Even though you do not know those details, by reading this at the y-axis, you should understand what's going on here. I said this 100% is kinase activity. Then which compound is stronger inhibitor? Yes, that should be this compound here, because if you add this compound here, kinase activity becomes less than 10%. So this is how you can quickly screen out which motif is important for this position. This is another SAR. SAR was structure-activity relationship, right? So if you can modify this position with a small structure, seems like those are okay. If you modify with this big structure, now you destroy those activity. That means that this position is not good position to add your linker. So this SAR is to improve your activity. Also, the information can be used to design your affinity matrix molecule later. So let's say the first SAR was focusing on this position and you found this structure as the best one. You can move on to the other positions. By taking the most active motif here, isopropyl group for this position, now it's possible to modify the other position and figure out which compound is more active than the other. Previous case was preliminary SAR, that's why one concentration and measured those percent inhibition. But in this table you can see this is IC50 concentrations. To get IC50, you need to test different concentration of the inhibitors, right? So this is dose-dependent experiment and got your IC50 numbers. IC50 number-wise, the higher the better or the lower the better? It should be the lower the better, right? So among this table, seems like this compound has the lowest IC50, 35 nanomolar concentrations. So 35 nanomolar can be quite strong binding, it implies. That's why maybe next step can be, even you can do protein small molecule complex. Once you have a very strong small molecule binder to the protein, now even that is possible to make a co-crystal of this small molecule to the target protein. Once you have this kind of a co-crystal structure, now it is possible to do structure-based drug design. That means by looking at those binding mode, you can design even better fitting molecules for inhibiting these proteins. Still, the next step is you need to confirm the selectivity of this molecule against either CDK1 or CDK2. Now here is one example of a target validation. We have studied before the cell-free or ex vivo system. In this case, you can take egg extract. This egg is from Xenophus. Xenophus is one kind of a frog. And egg extract has a lot of proteins inside there. And then you can add the DNA. This DNA is isolated from semen sperm. If you add the DNA into this system, this mimics cell division. DNA condensed in the middle, and microtubule is attached to the DNA and then ready to attract those DNAs. This is a normal status of a cell division mimicking, but here if you add CDK inhibitors, now everything is screwed up. CDK is an important checkpoint for cell division. That's why if CDK2 or CDK1 is inhibited, proper organization of DNA and proper organization of microtubule is all disrupted. So this is a phenotype we can see by adding CDK inhibitors here. This is normal status. This is inhibition of CDK effect. So if you inhibit the proper process of cell division, now DNAs are not properly condensed, and also microtubule is not oriented properly to bind to the DNAs. Here we can apply affinity matrix techniques. So this is the active molecules affinity matrix, and this is the inactive molecules control affinity matrix. If you incubate your cell extract with this affinity matrix, you can assume active protein can bind to this molecule and can be removed. When you remove that active protein, the phenotype shows almost similar to free molecule treatment. But if you treat with this control matrix, now the phenotype is almost similar to the normal. That means this control matrix did not remove active component. Only this active matrix removed active component. So next step is to add back that missing component to this matrix to recover this phenotype to normal phenotype. So in this case, when they added the CDK1, the phenotype is fully recovered to the normal. But CDK2 did not recover this phenotype properly. That means the relevant target of this molecule is CDK1, not CDK2. The same affinity matrix material can be utilized for pull-down experiment. So this is a control matrix and this is a positive matrix. Sometimes we see only one band or two band from affinity matrix, but in many cases, practically we can see many proteins bound to the matrix. Then it's important to rule out those non-specific binders. In this case, this is a control matrix, this is a positive one. Then you can see most of those overlapping protein bands are non-specific binders. But you can see here, one protein here. This is very strong only in positive matrix. This can be potentially real target protein. Also, this band is confirmed by antibody treatment, and anti-CDK1 showed that the identity of this band is a real CDK1. Now here's one question for you. Utilizing antibody confirming those identity of those protein, what do you call it? Is it southern blotting, northern blotting, or western blotting, or eastern blotting? So this is your quiz 1, and I will put up one question in IVLE, so you may give your answer in there. Okay? This was one example of a target validation. So if you have a potential multiple target, you need to figure out which one is really relevant, functionally related target protein of your small molecule. But now, let's think more general scale of target validations. If you have a small molecule for phenotypic change, then you take that small molecule and utilize it as bait to find out the target protein. So this was a target ID. And then the next step we say is a target validation. Now if you find out that this is a target protein, then you can tell which gene is making this protein. Then to confirm whether really this gene's function is related to this phenotype, one very general approach can be you knock out this gene and compare that effect with compound effect. Or if you need a temporal control, you can do maybe knockdown effect. So if you do knockout or knockdown, if that phenotype is similar to your compound, what can you tell? Your compound effect can be its inhibition of the protein, right? If it is activator, what approach you should take? If it is activator, maybe you can think knock-in. If your compound is activating that protein, you make more copies of that protein. They may have related phenotypes. So in this case, if you find new protein with a new function, then this protein you can call this is new biomarker. And this new biomarker can be related to specific disease. When you found your compound as a specific binder to this new biomarker, your compound can be probe or new drug candidate. So this whole procedure is important to confirm your compound's role and mechanism, and this is the so-called target validation.